DNA modifying enzymes and significance. DNA modification refers to changes or alterations occurring in DNA molecules during replication, transcription, repair, genetic recombination, epigenetic regulations. DNA modifying enzymes, the enzymes that affect change in the DNA chemical constitution or topology, are generally referred to as DNA modifying enzymes. Broadly, they can be grouped into two categories based upon the nature of modification performed, like composition modification or composition modifiers. These are the enzymes that change the chemical constitution and require net gain or loss of covalent bonds. Major composition modifiers include enzyme classes such as nucleases, methyltransferases, methylases and demethylases, phosphatases, kinases, polymerases, and ligases. Then topology modifiers, they change the topology of the DNA molecule and does not result in net gain or loss of a covalent bond. These enzymes are primarily involved in DNA replication. They are generally classified into two classes based upon the number of DNA strands cut by the enzyme in single reaction. Topoisomerase type 1 single strand cut and seal, and topoisomerase type 2 double strand cut and seal. An important DNA modifying enzymes are nucleases. This class of enzymes break the phosphodiester bonds of the DNA backbone. They produce single and double stranded breaks in the target molecule. In living organisms, they are essential machinery for many other aspects of DNA repair. Defects in certain nucleases can cause genetic instability. There are two main primary classes of nucleases. Exonuclease, they attack at the end of nucleic acid and usually remove just a single nucleotide or sometimes a short oligonucleotide. Any particular exonuclease, either the 3' end or 5' end or not both. BAL31 exonuclease 3 endonuclease enzyme belonging to the class of nucleases degrade makes cut or at internal site. A number of different specificity endonucleases exist. Some examples are S1 nucleases. They cleave only single stranded DNA, including single stranded nicks and double stranded molecules. Then restriction endonuclease. They function to defend the cell from invading viral force by bacteriophages. They mostly cleave dsDNA, but only at a limited number of square sequence-specific sites. This subcategory of endonucleases recognize and or cleave DNA at specific sequences, recognition sequences, or restriction sites, as compared to no sequence specificity in case of their other nucleases. The REAs are further categorized into four types, type 1 to 4, based upon their properties. Applications. Restriction endonucleases are used in the process of molecular cloning. Exonuclease 3 is used for generating single-stranded templates. Then deoxyribonuclease, a nuclease enzyme that can catalyze the hydrolytic cleavage of phosphodiester bonds in the DNA backbone are known as deoxyribonuclease. Based on the position of action, these enzymes are broadly classified as endodeoxyribonuclease, cleave DNA sequence internally, and exodeoxyribonuclease, cleave the terminal nucleotide. Unlike restriction, once again, unlike restriction enzymes, DNases does not have any specific recognition restriction site and cleave DNA sequences are at random replications. There is a wide variety of recombinant deoxyribonucleases shown, which have different substrate specificities, chemical mechanisms, and biological functions. They are deoxyribonuclease 1 DNAs, and endonuclease, which cleaves double-stranded DNA or single-stranded DNA. The cleavage preferentially occurs adjacent to pyrimidine residues. The major products are 5' phosphorylate bi-, tri-, and tetranucleotides. The major products are 5' phosphorylated bi-, tri-, and tetranucleotides. It requires divalent ions and for its activity and creates blunt ends of 1,2 overhang sequence. DNase 1 is the most widely used enzyme in cloning experiments to remove DNA contamination from mRNA preparation, to be used for cDNA library preparation, northern hybridization, RT-PCR, etc. The mode of action of RNase 1 varies according to divalent cation used. Some of the common applications of DNase 1 are eliminating DNA containing contamination plasmid from preparations of RNA, analyzing DNA protein interaction via DNA footprinting. Then nicking DNA prior to radio labeling nick translation, etc. Then deoxyribonuclease 2, DNase 2. It is a non-specific endonuclease with optimal activity of acidic pH and conserved from human to C. elegans. It does not require any divalent cation for its activity. DNase2 initially introduces multiple single-stranded nicks in DNA backbone and finally generates 3' phosphate groups by hydrolyzing phosphodiester linkage. This enzyme releases 3' phosphate groups by hydrolyzing phosphodiester linkage and creating links in the DNA backbone. DNase2 acts by generating multiple single-stranded nicks throughout, followed by production of acid-soluble nucleotides and oligonucleotides. The catalytic site of enzyme containing 3 histidine residues which are essential for enzyme activity. Some of the common applications of DNase2 are in the DNA fragmentation as molecular weight marker and in cell apoptosis assays. Ribonucleases, nucleases that can catalyze an hydrolysis of ribonucleotides from either single-stranded or double-stranded RNA sequences are called ribonucleotides. RNases are classified into two types depending on position or cleavage: endoribonuclease and exoribonuclease. Two common types of ribonucleases are ribonuclease A. It is an endoribonuclease that cleaves specifically single-stranded RNA at the end of pyrimidine residues. Optimal temperature for RNase A is 60 degrees Celsius. Then ribonuclease H, RNase H. It is a non-specific endoribonuclease that degrades RNA by hydrolytic mechanism of RNA-DNA duplex, resulting in single-stranded DNA. Then enzyme-bound divalent metal ion is a cofactor here. The product formed is 5' phosphorylated ssDNA. During cDNA library preparation from RNA sample, RNase H enzyme is used to cleave RNA strand of DNA and RNA duplex. Significance: RNase is important for RNA maturation and processing. RNase A and RNase H may play important role in initial defense mechanism against RNA viral infection. They can be used to remove RNA contamination from DNA samples. Then DNA polymerase. They are groups of enzymes that are used to make copies of DNA templates, essentially used in DNA replication mechanisms. These enzymes make new copies of DNA from existing templates and also function by repairing the synthesis of DNA to prevent mutations. DNA polymerase catalyzes the formation of the phosphodiester bond, which makes up the backbone of DNA molecule. Types of DNA polymerase. 1. This is a type of A or family A polymerase enzyme. Its main function is excision repair of DNA strands in 5' to 3', 3' to 5', and 5' to 3' direction. It also helps in the maturation of Okazaki fragments, which are short DNA strands that make up the lagging strand during DNA replication. Its role during DNA replication is the addition of nucleotides at the RNA primer as it moves along the 5' to 3' direction. Then proteolytic cleavage of DNA polymerase 1 resulted in fragment without 5' to 3' exonuclease activity known as a Klenov fragment. Then DNA polymerase 2. It belongs to type A or family A polymerase enzyme. Its major function is a 3' to 5' exonuclease activity and also restart replication after replication stops due to DNA strand damage. It is found in replication fork to help in directing the activities of other polymerases. Then DNA polymerase 3. This is the primary enzyme that is used in DNA replication belonging to family C or type C. It is responsible for the synthesis of new strands by adding nucleotides to the 3' OH of the group of the primer. It has 3' or 5' exonuclease activity, hence it can also proofread the errors that may arise during DNA strand replication. Then TAC DNA polymerase. It is a thermostable DNA polymerase 1 that was isolated from thermophilic bacteria, Thermus aquaticus. It is commonly used in PCR to amplify short strands of DNA. Due to its thermophilic nature, it can withstand protein denaturation that is required during PCR. Then decay. DNA ligase catalyzes the formation of phosphodiester bond between two deoxynucleotide residues of two DNA strands. DNA ligase enzyme require a free hydroxyl group at the 3' end of the DNA chain and a phosphate group at the 5' end of the other and require energy in the process. The role of DNA ligase is to seal nicks in the backbone of double-strand DNA after lagging strand formation to join the Okazaki fragments. This joining process is essential for normal synthesis of DNA and for repairing damaged DNA. It has been exploited by genetic engineers to join DNA chains to form recombinant DNA molecules. The most widely used DNA ligase is isolated from T4 bacteriophage. T4 DNA ligase needs ATP as a cofactor. The enzyme from E. coli uses cofactor NAD. Except this, the catalysis mechanism is somewhat similar for both the ligases. Then discontinuity repair, discontinuous DNA ligase joining two molecules, DNA ligase significance. DNA ligase enzyme is used by cells to join the Okazaki fragments during DNA replication process, used in repair of discontinuity, a missing phosphodiester bond in one strand, joining two molecules of dsDNA together. Molecular cloning ligase enzyme has been routinely used to construct a recombinant DNA. Then methyl transferase methylase. Methyl transferase or methylase catalyzes the transfer of methyl group to its substrate. The process of transfer of methyl group in its substrate is called methylation. Methylation is a common phenomenon in DNA and protein structure. So methyl transferase uses a reactive methyl group that is bound to sulfur in S-adenosyl methionine, which acts as a methyl donor. Methylation normally occurs on cytosine residue in DNA sequence. In problem, methylation occurs on nitrogen atom, either on N-terminus or on the side chain of protein. So DNA methylation regulates gene or silence gene without changing a DNA sequence as a part of epigenetic regulation. In bacterial system, methylation plays a major role in preventing their genome from degradation by restriction enzymes. It is a part of restriction modification and system in bacteria. Restriction enzymes cleaves within the recognition sequences if the DNA is unmethylated. On methylation by methylases, the restriction enzymes are inhibited by cleaving within the restriction site. Then significance: inhibit binding of transcriptional machinery protein to DNA after blocking gene expression, recruit specific repressors at site that then switch off nearby genes, gene silencing by recruiting histone modifying enzymes, allows discrimination between parent and daughter strands during DNA repair, maintenance methylation, then protect DNA from action of endonuclease, which are parts of restriction modification system. Then phosphatases. Phosphatase catalyze the cleavage of phosphate PO4-2 group from substrate by using a water molecule hydrolytic cleavage. This reaction is not reversible. This shows totally opposite activity from enzyme like kinase and phosphorylase and add a phosphate group to their substrate. On the basis of their activity, there are two types of phosphates, acid phosphatase and alkaline phosphatase. Acid phosphatase, it shows an optimal activity at pH between 3 and 6. Lysosomal enzyme that hydrolyzes organic phosphates liberating one or more phosphate groups. They are found in prostatic epithelial cells, erythrocyte, prostatic tissue, spleen, kidney, etc. Alkaline phosphatase. It is an homodimeric enzyme that catalyzes reaction like hydrolysis and transphosphorylation of phosphate monoester. They show their optimal activity at pH of about 10. During post-translational modification, alkaline phosphatase is modified by N-glycosylation. It undergoes a modification through which uptake of 2 Zn+2 atom and 1 Mg2 plus ions occurs, which is important in forming active site of that enzyme. Alkaline phosphatases are isolated from various sources like microorganisms, tissue of specific different organs, connective tissue of invertebrates and vertebrates, and human body. Then polynucleotide kinase. This is a homotetramer with phosphatase activity at 3' end and kinase activity at 5' end with a tunnel-like active site. Polynucleotide kinase catalyzes the transfer of a phosphate group from Y position of ATP to the 5' end of either DNA or RNA and nucleotide monophosphate. PNK can convert 3' PO4 5' OH ends into 3' PO4 5' PO4 ends, which blocks further ligation by ligase enzyme. PNK is used to label the ends of DNA or RNA with radioactive phosphate group. T4 polynucleotide kinase is the most widely used PNK in molecular cloning experiments, which was isolated from T4 bacteriophage infected E. coli. Then DNA glycosylases. These are the enzymes that generally recognize the damaged base nucleotides present in DNA and remove them. They hydrolyze the glycosidic bond between sugar and nitrogen base, producing a basic site in the DNA. They are one of the key components of general DNA repair system. Then topoisomerases. These are the enzymes that change supercoiling topology of the DNA molecule. They generally remove supercoils preventing relieving the torsional stress pressure present in the overwound DNA during replication. They constantly break the DNA backbone, thereby allowing the DNA supercoils to relax. Topoisomerase is classified according to mechanism they use for changing DNA topology. Type 1A and 1B. They break one of the two strands of DNA, rotate the end of the broken strand around the intact strand, and then seal the ends. The reaction does not require ATP. Type 2, they cleave both strands during reaction, rotate both ends by 360 degrees, and then reconnect the respective ends. The reaction is powered by ATP hydrolysis DNA gyrase of E. coli.